The overall goal of this procedure is to generate left intrastriatal brain hematoma formation in a mouse. This is accomplished by first anesthetizing the mouse and mounting the head in a stereotactic frame. The next steps of the procedure are to expose the skull and drill a burr hole. Then, the left striatum is injected with collagenase, or blood, and the wound is closed up. Ultimately, worsened neurobehavioral function can be detected by rotarod testing, and the hematoma formation can be detected by magnetic resonance imaging and hematoxylin and eosin staining.
Intrastriatal collagenase or autologous blood injection can help answer key questions in the field of intracerebral hemorrhage, such as the effects of hematoma expansion, cerebral edema formation, and secondary injury mechanisms. These techniques allow in vivo testing of promising therapeutic interventions aimed at secondary injury mechanisms after intracerebral hemorrhage, at the same time that discovery of these targets occur and transgenic systems are created. Generally, individuals new to this method may initially struggle with mouse intubations, drilling craniotomies, intrastriatal injections without leakage, and appropriate post-surgical care. Visual demonstration of these models is critical as highly reproducible injury is essential to in vivo study of pathophysiology and interventions. There are many critical steps that require experience to avoid error and subsequent production of dissimilar injuries. Demonstrating the collagenase injection procedure will be Beilei, a research scientist in the Multidisciplinary Neuroprotection Laboratories. Demonstrating the autologous blood injection procedure will be Huaxin Sheng, an assistant professor in the Department of Anesthesiology at Duke University.